For abstracts of redox reaction and catalyst activity. Redox reaction is a chemical reaction in which the reactants undergo a change in oxidation state. All the redox reaction can be broken down into two different processes, which is reduction and oxidation. A catalyst is a component that accelerates the chemical process while not being consumed by it. The catalyst test is done by placing a drop of hydrogen peroxide onto a microscope slide. An applicator stick is touched to the colony and then the tip is mirrored onto the hydrogen peroxide drop. If the mixture produces bubbles or froth, the organism is said to be catalyst positive.
Redox reactions are essential for cellular process, energy transfer, and chemical transformation, while catalysts and enzymes protect cells from oxidative damage by breaking down hydrogen peroxide into water and oxygen. This laboratory experiment aims to explore redox reaction and catalyst activity, understanding factors influencing reaction like substrate concentration and temperature.
Next, I'm going to tell you the methodology. We have three parts. The first one is observe a redox reaction. 3 pieces of apple were exposed in different environments. First one was we exposed to the air, second we distilled water, and the third one with the lime juice. Wait for 30 minutes and the color change was observed. Next, the catalyst activity in living tissue. 3 grams of 4 different samples, which are liver, potato, apple, and soap piece were inserted in the different test tube. So the reaction line was marked and 5 ml of 3% hydrogen peroxide was put in each test tube. Wait for 5 minutes and observe the bubble formation. The third one is the effects of temperatures on catalyst activity. Use 3 pieces of potato with same weight, put them in a different test tube and mark the starting line for the reaction. 5 ml of 3% hydrogen peroxide was put and each of them were placed in a different temperature with ice bath, room temperature and water bath with 0, 24 and 34 degrees Celsius respectively. 5 minutes were wait and the result was observed.
The results of observing the redox reaction are apples that are exposed to air and distilled water change color to brown, while the ones that are exposed to lime juice do not change color. Next, the results of catalyst activity were determined by the rate of bubble formation after 5 minutes, which both peas and apple were low, potato was medium, and chicken was high. For the last experiment, the one that was put in an ice bath had low bubble formation. At room temperature, it had medium bubble formation, while in a water bath, it had high bubble formation.
For conclusion on this experiment, the observable result of enzyme functioning as an excellent biological catalyst is redox reaction. It was noted that when the medium was exposed to air, distilled water and lime juice, the color changed. It was discovered when the amount of gas bubble produced by potatoes, apple and chicken flesh corresponded to the level of catalyst activity detected on living tissue. It was also noted that the temperature had an impact on catalyst activity.
Experiment aims to identify differences in respiration rates and metabolic activities when 20% solutions of each carbohydrate are introduced to yeast cells. The experimental setup involves yeast cells and solutions containing 20% glucose, lactose, or maltose with BTB as an indicator. BTB changes color in response to alterations in pH, allowing for visual and quantitative assessment of metabolic activity. The spectrum photometer is employed to measure the absorbance of BTB at specific time intervals, providing a quantitative metric for the rate of cellular respiration.
Cellular respiration is a biochemical process that organisms use to extract energy from their organic compound. This laboratory experiment aims to investigate the cellular respiration rate of yeast cells using different carbohydrate substrate, which is 20% of glucose, lactose, and maltose. This experiment employs a spectrophotometer to measure change in absorbance, providing insight into the metabolic activity during the breakdown of the sugar.
The full methodology for cellular respiration, firstly, we will add 5 ml of 20% glucose, 20% maltose, and 20% lactose into each test tube A, B, and C, respectively. Next, we'll fill the test tube with 10 ml yeast suspension and transfer it to each of the test tube A, B, and C. After adding yeast suspension, we will add 3 drops of bromo-thiamal blue indicator into each test tube and shake gently to make sure it is distributed evenly. After that, we'll place the test tube on the test tube rack around 5 minutes before observing the color change. Next, we fill a cuvette with the reaction mixture from each test tube and wipe the outside of the cuvette with clean wipes to make sure it is clean. Lastly, we will measure the absorbance at a suitable wavelength, which is 570, using a spectral photometer.
As you guys can see from the table, the result that we obtained, we measure the absorbance on each solution. Firstly, the absorbance for bromo-thiamal blue indicator is increases. Secondly, the absorbance for 20% of glucose plus yeast is decreases. Thirdly, the absorbance for 20% of lactose plus yeast is increases. And last but not least, the absorbance for 20% of maltose plus yeast is decreases.
Overall, the experiment showed that yeast prefer glucose for energy source, while lactose and galactose are less efficient. Qualitative measurement for cellular respiration can be measured with a spectrophotometer. That's all. Thank you.